Hello everyone, in today's video we'll be discussing about mitosis. So mitosis is a part of cell cycle which helps the cell to divide, and in the end of mitosis we get identical copies of cell. Okay, so let's just before starting let's just uh, uh, discuss about the key points of mitosis. So every cell wants to make a copy of itself, and for that to happen, they undergo cell cycle. And the cell cycle is divided into various stages. We'll not be discussing about cells, the G1 stage, S phase, and G2 phase in the video. There'll be an upcoming video about that. In this particular video, we'll be discussing about the M phase or the mitosis phase of the cell cycle. Okay, so in the mitosis, the cells which we get in the end are identical in nature, and the number of chromosome before and after the mitosis remains same. Will be particularly this is the key to the mitosis, and we'll be discussing about this sentence, and we'll discuss about this in detail in this video, and I'll explain how the number of chromosome before and after mitosis remains same. um, and mitosis occurs in somatic cell. The basic purpose of mitosis is to increase the number of cell, and it helps in increasing the cell count. And if a cell is damaged, to repair the damaged cell, it undergoes mitosis. Okay, so it's not involved in any kind of sexual reproduction. Okay, so before discussing the stages of mitosis, I just want to clear a basic and the most confusing terms for students. Okay, so a cell before entering the mitosis phase appears like this. It's having a cell membrane. There's a double membrane, nuclear membrane, and there are thread-like structures which we call chromatin. Here we cannot see the chromosome at this stage. We under microscope we are not able to see the chromosomes because they are not in condensed form yet. Okay, so we are having these thread-like structures. So is ki, what happens is students are basically confused. So uh, if I asked you to draw a chromosome, okay, so if I generally ask students to draw a chromosome, what they draw is this. They'll draw this. This is incorrect. Even though this is a chromosome, but it's a duplicated chromosome. Let me explain. Okay, so if someone asks you to draw a chromosome, you're supposed to draw this. Okay, you're supposed to draw this is a chromosome, and let me draw the centromere for you. Okay, so this is a chromosome. So after the DNA replication, okay, after it the cell undergoes S phase and the DNA is duplicated, it becomes like this. It becomes like this, and this is what people usually associate with chromosome. Yes, it is a chromosome, but this chromosome is duplicated. That is it. This is actually pairs of chromosome. Okay, so here we the okay, let me explain again. This is what a chromosome is. So whenever someone asks you to draw a chromosome, you're supposed to draw this. And this is a duplicated chromosome, and it appears after the S phase, that is after the DNA replication has taken place. Okay, so now the black dot you see over here, it is um, it is the centromere. This is okay, this is centromere. And what are sister chromatids then? These two, this and this, these are sister chromatids. Okay, so what is a chromosome? This is a chromosome, and this is a duplicated chromosome. Here you can see the black dot represents a centromere, and these are sister chromatids. Okay, so now let's move on to the S phase. So in this mitosis is divided basically into four stages: prophase, metaphase, anaphase, and telophase. One by one, we'll discuss what happens in what stage. Okay, so this the cell which I showed you earlier, uh, this cell, this this is how cell appears in the interphase or before mitosis. Okay, so in the prophase, which is the first stage of mitosis, the nuclear membrane disappears. As you can see here, there was a nuclear membrane over here. Okay, in prophase it disappears and chromosomes become visible. So the X-shaped structure which you can see now you can identify the chromosomes at this stage. So whenever you're making an onion root slide and the teacher or your professor is asking you to identify the prophase, so what you see here, now you cannot see the nuclear membrane and you can see the condensed structure. Now you can see the X-shaped structure of a typical chromosome, a typical duplicated chromosome or replicated chromosome. Okay, so in prophase the nuclear membrane disappears and chromosomes um, appear because they are now condensed, they're in condensed form. Okay, this stage is metaphase. Here you can see that all the chromosomes now are aligned at the metaphase plate or the equator, and you can see that the centromere is linked to the spindle fiber. Okay, so in metaphase the chromosomes align at the equator. In the and this stage is the easiest to identify under a microscope. All you just need to see is the chromosome have to be at the equator of the cell. Once you identify that, you can easily say this is this metaphase of mitosis. In the anaphase, the sister chromatids now are separated. Here you can see that the sister chromatids are attached. They are separated now, and this sister chromatid now would be considered a full-fledged chromosome. So if the question appears in the question paper, if they are asking, suppose they have given you the diagram, now they are asking you identify the number of chromosomes. So you'll say 4. And now they are asking you to identify the number of chromosome in this stage. So you'll identify the stage that this is anaphase, since you can see that the sister chromatids are moving towards the opposite poles. So here the number of chromosome would be 8. So to prevent any kind of confusion, whenever someone is asking you to count the number of chromosome, you simply count the number of centromeres. So here we are having 4 centromeres. So 1, 2, 3, 4. So we have 4 chromosomes. Now here you are having 8 chromosomes. How? 1, 2, 3, 4, 5, 6, 7, 8. Okay. So let's just revise. This is prophase. Here the nuclear membrane is disappeared and the chromosome appears. By that I mean they are condensed and now they are visible under microscope. Okay. So in metaphase, the chromosomes are aligned at the equator. In the anaphase stage, the sister chromatids separate and they start moving towards the opposite poles. Okay. The next stage is telophase. Here you can see the nuclear membrane is reappeared and the sister chromatids, now called chromosomes, they have arrived at the opposite poles. Okay. So now here at this stage, we'll not call them sister chromatids, we'll call them chromosomes only. Okay. So in telophase, the nuclear membrane reappears and the chromosomes are at the opposite poles. And after this, after cytokinesis, that is the cytoplasm also divide and we get 2 daughter cells. The 2 daughter cells are identical. Here you can see there are 4 chromosomes. Here also there are 4 chromosomes. Okay. So after DNA, okay, this cell now will further undergo S phase and the DNA duplication will take place, and again when it undergoes prophase, it'll get this look of duplicated chromosomes. Okay, so in this video, so far we discussed what is mitosis. So mitosis is the part of cell cycle which results in identical cells. The number of chromosomes before and after the cell cycle is always same. Remember, in the beginning I told you guys that the number of chromosomes before and after mitosis remains same. So let's check if the number of chromosomes before and after mitosis is same. Here we are having 4 chromosomes, correct? In the prophase. Here we are also having 4 chromosomes. So the number of chromosomes before and after mitosis is same. Okay, let's just summarize the whole mitosis. This table, you can take a screenshot of this if you want. This table will help you to memorize the key events happening at the stages of mitosis. So the first stage of mitosis is prophase. Here you can see the nuclear membrane is broken down, okay, and the chromosome is attached to the microtubule of the spindle fiber. This you cannot clearly see in under the microscope most of the time, okay. So the next stage is metaphase, and this is the easiest phase to identify under microscope, okay. Why? Because in this stage the chromosome align at the middle, okay. The next stage is anaphase. The sister chromatids are separated and now they move towards the opposite poles. The next stage, telophase, now here we were calling them as sister chromatids, now we are calling them chromosomes because now they are separated, okay. So they have moved to the opposite poles and the nuclear membrane reappears, okay. That's it. I hope you like this video and I hope it helped you in certain way. And if it helped you in any way, please give it a thumbs up and ask your doubts in the comment section. Please ask any doubt in comment section. Okay, thank you for staying. Goodbye.